Our next speaker is Dr. Ashvin Patel. He will be speaking to us on Phoenix diffusion system design and use. Dr. Ashvin Patel is the Director of Analytical Research and Business Development at Teledyne Hansen Research. He has over 25 years of pharmaceutical industry experience, mainly in analytical chemistry. Dr. Patel, you have the mic now.
Thank you, Rong. Just to check, can you hear me well?
Yes, we can see
Okay,
your slide and we can hear you clearly.
Thank you so much. Thank you so much for kind introduction. I'd like to say that in past 2 days, we all have learned a lot on IVRT and IVPT about the science, the requirements, and the techniques behind the diffusion studies. So in this presentation, I have not included or mentioned anything about the theoretical aspects that we already discussed. So many experienced, knowledgeable speakers has mentioned so many things about it. So I have skipped that one. And as an equipment suppliers, I can say that they can innovate and bring improvements to the test, discrimination, increase efficiency, safety, etc. And I'd like to show the feature and the new advancement from an equipment manufacturer's perspective, which will benefit end users and the entire pharmaceutical industries. So here, this is our previous design of Vision microarray system, which is discontinued. And that Vision microarray system was the first automated and patented design came out from Hansen. However, there were some issues, some glitches with it, which we learned over a few years. That is the difficulty to measure the membrane temperature prior to starting the test, time consumption in the cell preparation, pre-wetting or conditioning of the membrane needed to be performed outside the cell body. Perhaps the other system available in market today has also that requirement. The dosage is applied on the membrane outside the cell body. So possible time difference between cell 1 to cell 6 from sample application and starting the test. This is because the dosing is done outside the cell body and the test is started once all 6 samples are prepared and mounted on the system. And that also comes with the experience and length of the experience of the person who is working and the training. Stirring required to stop during the sampling to avoid sample dilutions. So in the Vision microarray system, if stirring remains continue and sample starts to pull because it's replacing from the bottom, pulling from the top, there is a chance of sample dilution. That's how there was kind of a little tricky thing to do. So stirring was stopped. And mixing is playing a vital role in homogeneity, which can lead to inconsistencies in RSDs. And due to the design, the exposed membrane orifice was inconsistently applied to each cell and possibly causing alignment and surface area exposure inconsistency, which also leads to somewhat higher RSDs. So now why and what have we improved? Based on users' feedback and the industry expert like Dr. Kailash Thakkar and the Turgis Pharma IVRT team, because we are equipment manufacturers, so we rely on feedback from the users and some experts. So in order to meet the regulatory requirements, such as a total system equilibration, including the membrane equilibration, membrane temperature measurement prior to dosing, accurate measurement and control of sample amount, which is applied to the membrane, frequent temperature and speed recording throughout the study. To reduce the variability, obtain consistent results with low RSDs. Better homogeneity of the receptor solution, as you have learned or heard from many speakers that samples should be collected from the center, not from the arm. There is no homogeneity in the arm and whatnot. So that's what we have improved and brought it in very highly efficient mixers. So we have also removed the need of stop stirring during the sample collection. That means the test, once it started, stirring will stop only at the end. And we created a user-friendly apparatus for ease of use. 21 CFR part compliant modules we have come up with. And Teledyne Hansen is always a state of innovative, is in a state of innovatives, and that is our business model. So we always innovate, but we need feedback and we need some support from industries in terms of their opinion, and then can lead us for modifications. So the result of our cell innovations. So what we have done like a multiple things and then come up with the patented Phoenix diffusion cells, which works with synthetic and biological membranes, as all the other diffusion cells are also working with that. Cell caps made on a CNC machines with uniform and high precision orifice. You will see like in some next slides about that. Cell components can be easily customized upon request. So if somebody thinks that they need a different orifice size for study, Hansen can definitely do that. So exposed membrane surface area is perfectly matted with the cell body each time. So there will be no alignment issue. Outside the cell is precisely made to ensure the flush contact with the heat source, because we have learned that maintaining the temperature is very, very important and critical. So this new technique is definitely going to maintain the temperature very, very well within the limitations, the restrictions or the specifications needed for that study. The membrane wetting and whole system conditioning occurs directly in the cell body. So you mount everything and you wait for the temperature to reach to the desired level, and during that time, system will be conditioned, including the membrane. So here is our Phoenix systems vertical diffusion cell design. And you can see on the left, we have marked completely like the different portions of that one. And what we have done here, you can see that this is a fill mark. That is very important for every user to understand and see that they can fill up to the fixed volume once they have set with that cell mark. Those cells are made from a precision bore cells made from a syringe body. It's with a more accurate than the hand blown glass. So cell to cell sample volume, cell volume variation is very, very less. It's a wide sampling arm, you can see here, than the all previous systems. And it's maintaining the homogeneous receptor solution throughout the cell. And on the right here, you can see we have three different kind of cells, small, medium, and large. And we have two different type of mixers, tall and short. So you can see that in the next slide that what it can benefit the users. Here is flexibility in study design. So we have a whole range of the components which can provide complete flexibility to the end users. You can see the membrane size is 25 millimeter. Cell volume ranges from 10 ml to 30 ml. RFS diameters are 9 mm to 20 millimeter. And I can say here something about the orifice diameters are well controlled within plus minus 0.1 millimeter or less. Dosage volume can range from 0.25 to 6.2 ml, depends on what type of the dosage chamber you guys are going to use. The dosage wafer here as what you can see on the left, and then you can see here on the right. And then we have specially designed threaded caps for the skin application, because most of the times with this type of dosage wafer, skin will not be remain in place and it will not be in contact. Many times it can come out from the receptor contact and whatnot. So that's how we have specially designed these threaded caps. So now here what I was talking about, the full spectrum, the full range of the customization, or the users can implement these in order to get their full diffusional sink conditions with solubility and what not the other presenters have discussed so far. So it's for an example, if you take a small cell which has a 10 ml volume, it can give you like a two different orifice sizes, one 9 millimeter and 11.3 millimeter. The membrane area is like 64 millimeter square to 100 millimeter squares. And the dosage height is 4 mm and 20 mm. That dosage height is going to give you that dosage volume of 0.25 millimeter to 1.3 millimeter. So as some products, it's like very low solubility, or some of them are very high soluble. And that's how we have come up with these based on the industry need and feedback what we got from them. And I recommend here something that the exact cell volume for each cell to be measured in the laboratory and use that volume in calculation. Like if you get 10.04 ml instead of 10.10 ml as what we have said here as a typical cell volume with the mixer. Because we have two different mixers, tall and small, definitely we recommend that cell volumes should be measured in the lab, and that is a one-time time investment and data collection and recording, and each components are serialized, so that will be very easy to record and to use for all subsequent calculations. It's a Phoenix dry heat system, like Phoenix, Arizona, dry heat. That is nothing else. No heat, water circulating or anything. It's a computer-controlled heating and stirring blocks for precise speed and temperature. So temperature and speed limits exceeds USP 1724 requirements. Typically, we obtained 0.5 degrees C versus the 1 degree centigrade required by USP, and speed is plus minus 2% versus the 10% requirement from USP 1724. It is a 21 CFR Part 11 compliant software which is provide data integrity. So very easy to detect and remove the bubbles, as you have heard from many speakers about the bubble and what it can cause the problem, and you must have experience about having bubbles give you trouble and then you have to repeat the study and whatnot. But in this system, it is very easy. You can see in some of our video how we are showing that how to detect and remove the bubbles. Now this is our 6-cell manual sampling system. It's a very small footprint. You can see it's not going to consume very much space on a lab bench, because most of the lab are jammed with so many instruments and always lack of space in the laboratories. It's easy angle for the proper sampling, and media replacement with pipetter, or even a syringe can be used here. Stirring speed and cell temperatures regulated by a programmable stirring and heating block. So this one is a stirring and heating block. And the block, this one is controlled by onboard computer that is built into this touch screen display, and ability to print the parameters as desired. So whatever the test parameters we set and we conduct the test on it, it is going to saved and it can be printed as well. This is our robotic diffusion station, which is an automated sampling system. And some of presenters have already shown these pictures. I would like to just mention about that one, that it has that XYZ drive. That means it is going to take this needle and set at the proper placement, when to replace, how to rinse, and how deep it should go into the cell body and all the stuff. But that can be done at the time of installation. So once it's installed and qualified, no need to change any of the parameters, because for the small, medium and large cells, those parameters are fixed and all of them can be done at the time of installation. It has this sampling arm. This is available in a PEEK or stainless steel both metals. You can use it whenever it's needed based on the compatibility of the material which we are working with. It has a robotic arm. It moves automatically as per the program. These are the diffusion cells. These are the heating blocks here. And it can contain 6 cells on one side, 6 cells on another side, and total 12 cells it can hold. It has a wash station here. You can program it into the software saying that, okay, before or after each sample, I want to rinse this sampling arm with whatever the solvent is being taken from outside from an external source. And it can replace the media from those bottles here. And it can be programmed. If you want to do like a pure research, you want to do n is equal to 3, you can do easily 4 experiments into one setup, and you can have flexibility to replace from all 4 different media bottles here. It's a fully programmable systems which allows unattended testing. Advantage for users, I can say that our system is very user friendly. Prepare and collect the samples either manually or automatically using the software. Membrane temperature can be measured using IR thermometer, as what we have discussed that conditions is required to maintain the temperature, measure the temperature and maintain the temperature. So these can be done using an IR thermometer. And also the heating block temperature and the stirring speed is always going to be recorded per the time interval we define into the protocol or into the method on the software. It's a staggered dosing and starting of the test. And same way the sampling is going to happen precisely in a timed fashion. So even if you use the manual system, it is going to alert that it's time to sample from cell 1. Or when you start, it's going to say that, okay, it's time to dose the sample 1 or dose the sample 2, as you define the time interval between the sample preparations. It's a very wide sampling arm, which is creating homogeneous solution, as I said before. So bubble detection and elimination can be done very quickly and very easily. It's like less than 2 seconds or something, you can definitely see and even remove the bubbles from that. Easy to control the dosing of the product. 21 CFR part compliant software. Test protocols can be saved, reviewed, and archived. This is for like a quality assurance, quality control, and regulatory perspective. You can have the test report printed and it can be saved. It is always going to be saved if it's programmed properly. Anytime the test history is available for verification and review, and all events and errors, if anything occurs, they are going to record it into the electronic log. So I'm going to do like a small video demonstration. There are 3 videos here, and one is going to talk about how to prepare a cell for IVRT. The second one will be how to prepare a cell for IVPT, and overall the system video, how the whole function is working. So I'm going to play that video in the next slide. To demonstrate how to set up a cell for
Dr. Patel, we couldn't hear the sound.
Okay, so bear with me for a moment.
Dr. Patel,
Okay,
looks
so
like the video is freezing right now.
yeah, so what I'm going to do, I'm going to talk over as the steps is shown in the video. So this is the demonstration of how we are going to prepare the cell for IVRT, as the membrane is going to taken as a dry, and then is going to put at the bottom portion of the cell cap, and then top portion of the cell cap is locked in place and secure the membrane there. Now he's going to fill up the cell with the receptor solution. And this membrane the cap is going to be placed on it and then tilted the cell for removal the air bubble and to fill it properly. So now by tilting this, you will not see any air bubble, and the cell is going to be completely full when the media is passed, the meniscus is passed through the center dot. That is the fixed volume with that one particular stirrer, and then it's going to be placed on a cell block. We're going to wait for the temperature to raise to 32 degrees or the desired temperature. Once it's reached desired temperature,
restart your video. Your video isn't moving forward.
It's going to be measured using the IR thermometer and going to be recorded the temperature. Once the temperature is there, we can begin the sampling for dosing the cells.
Ashwin?
Yes.
It's a little hard to follow because the video is also not playing. I think everything is working, but when you share it in WebEx, you may just want to, when you get the big black share window, you may just want to, in the top right, click that little X on the speaker there, and then it may play. Do you want to give that a try?
Okay.
You can stop sharing and then reshare.
Stop sharing.
And then when you reshare at the top right, where it says automatically optimized, there's a little speaker to the right of that. Has an X in it. So just take that X away and then give it a try again, just playing the video.
Sure, I'm going to do that right now. And
then you can start the video again if you'd like.
Yes.
I'm going to demonstrate how to set up a cell for an IVRT test. So we're going to take our diffusion membrane and place it in the bottom portion of the cell cap. And then we're going to take the top portion of the cell cap and put it on to secure the membrane in place. Next, we're going to take our receptor solution and fill the cell. We'll put our cap into place and we'll tilt our cell to fill the rest of the way. The cell is fully filled with media when the meniscus passes through the center of the white dot on the sampling arm. Once it's assembled, we can put it into its position on the cell block. Now we're going to allow the receptor solution and membrane to equilibrate to the desired temperature. Once it's reached the desired temperature, we're going to take an infrared thermometer to measure the surface temperature of the membrane. Once we see that it's at the temperature, we can begin to prepare the sample. So I'm going to take our sample and apply it into the dosage chamber to the membrane. We will then take our spatula to remove any excess and remove any bubbles from the dosage chamber. Now our sample is fully applied. We will take a clear dosage lid and cover our sample. Now we're able to start the test and repeat the process for all cells.
So now I'm going to play another video.
I'm going to demonstrate how to prepare a diffusion cell for an IVPT test. First, we'll take our skin and gently stretch it. And place it on the base of our cell cap. We'll put the skin and insert into the bottom portion of the cell cap and secure them with the screw cap. Next, we'll fill our cell with receptor solution. Then we'll place our cap into position. We'll tilt the cell to remove any air bubbles and fill the cell with receptor solution to the white dot on the sampling arm. The meniscus of the receptor solution should pass through the center of the white dot. We're going to repeat this process for all the cells being used, and we'll place it back into the cell block. Now we're going to allow the receptor solution and skin to reach physiological hydration and temperature conditions. We'll measure the temperature of our skin using an infrared thermometer. We will conduct a barrier integrity test on the skin, such as a TEWL measurement using a vapometer. Once the skin passes the requirements, we're ready to apply the sample. To apply the sample, we're going to use a positive displacement pipette. Using a flat surface, such as an HPLC vial insert, we're going to ensure that the sample is evenly spread on the skin membrane. Then we're going to determine the amount of sample applied using an appropriate weighing method. And we'll repeat this process for all 6 cells being used.
Okay, so I would like to mention a few things here on this video that for demonstration purpose, we have generated this video very quickly into our lab. Do not use any sharp forceps as what we have used here because it was easily available and handy. So we have used that. And for various type of positive displacement pipetted tips can be used for different viscosity or different thickness of the product. And we have used the gel product on which we are currently working on. So those few things I just want to mention it. And the weighing techniques, what we have talked about it here in the video is like you can pre-weigh and post-weigh and get the exact weight, including those HPLC vial inserts, which is used to spread the product. So if anything is going to stick to that surface can be accurately measured and we know exactly how much amount is applied. So I'm going to, I know we are running little late, but there is another video which I want everybody to see it and then understand, and I may speak over that video because that is taken from our website. Some of you might have seen it, but I would like to take this opportunity and play it here. This is our entire system. And you can see the sampling was done. This is for homogeneity. It's a quickly mixing. The protocol creation is very easy. The sampling trays, the HPLC vial tray can be placed on directly HPLC once all samples are collected. There is another way of how the cells are being prepared. But the main technique is going to remain same as place the dry membrane, secure it, fill it with the receptor solution. And how can you fill it? It's all the different, you can do it. And then you have noticed that there is a small air bubble remain, but it's going to show in the next video somewhere that how to detect and remove that one. it's an instant homogenization of receptor media. So this is for bubble detection. You can see the bubble here. It's also there in other cell, but this is the one how it's going to be removed. This is a sample application. I would like to mention that when you apply the sample, do not use excessive force, which is going to lead the product to diffuse through quickly or alter the product, alter the results in your IVRT studies. It's a touch screen display with the color display, so it's easy to see it. And very easy interface to use it. So it's talking about second sample. It's going to collect the sample in 14 seconds. This is the movement of the robotic diffusion system cells. The arm is going to move and reach to the cell as programmed or as needed for the protocol. And this is rinsing. This is how to create the protocol. It's a very easy, and you can have all the information needed for that particular test. It's a very large database, so there is no need to worry about when it's going to get full and then when we have to do some kind of archival, but definitely based on the requirement for individual companies and the regulatory bodies request, the data can be archived and stored and saved securely. It's 108 vial capacity. And that test report looks like this. It has all the parameters what we have entered, and then this is the recorded temperature and stirring speed. And this temperature and speed, the temperature is coming from a heating block. But once all the cells are maintained and then heating block temperature remains, all cells are going to be at the same temperature as what we have initially measured. You know, it's an unattended operation. It can be done overnight and sampling can be done when we come in the next day morning, because typically we are seeing test is done for 6 hours, up to 6 hours and longer. So this automated system is really helpful for everyone. Here, I conclude with this by thanking everybody. Like on behalf of Teledyne Hansen Research, I would like to thank organizers and US FDA and all those who have supported for this opportunity to present on this webinar. And thank you all for attending the session. And if you guys have any question or any suggestions, you can send an email to me. It's listed here, or you can go on Hansen Research website and under technical support request. And this is our physical address of the company where all instruments are being manufactured in the United States. Thank you all.